So hello, I'm Mridul Mohan from group number 324. I'm here to present two topics, and the first one is deciphering of the genetic code and its meaning. So the textbook is the Biophysics, written by M.V. Voxen. So let us start. The first topic is codon degeneracy. The genetic code is a degenerative, meaning multiple codons can code for the same amino acid. This degeneracy is particularly evident in the third position, a small z, of the codon. Many amino acids are specified by the codons that differ only in the third nucleotide. Second one is the codon structure and the interactions. The codon is composed of three nucleotides X, Y, and Z. The first two nucleosides X and Y are more critical for determining the amino acid than the third nucleotide Z. Codons can be divided into two sides based on the composition of the X and the Y nucleotides. Codon and anticodon interaction. The third topic. The codon interactions with an anticodon on tRNA during translation. The interaction between the first two nucleotides X and Y of the codon and the corresponding nucleotides on the anticodon is stronger and more specific than the interaction between the third nucleotide Z and the anticodon. The weaker interaction at the third position allows for a verbal pairing where non-standard base pairing can occur. The table 8.4, it figures out, this table summarizes the genetic code. Each codon is represented by the three letters, example UUU and UUC, etc. The first two letters X and Y determine the amino acid, while the third letter Z often has less influence. So, in table 8.5, the formation of pass by the third nucleotide of an anticodon is represented. It shows the possible base pairings between the third nucleotide of the anticodon Z and the third nucleotide of the codon Z. The letter I represents the minor nucleotide iosine, which can pair with the multiple nucleotides. Genetic code and protein structure. This is a main topic in this section. The genetic code is a set of rules that translates nucleotide sequence in the DNA or the RNA into amino acid sequence in proteins. The primary structure of protein determined by its amino acid sequence dictates its three-dimensional structure and ultimately its function. So, concluding by the interaction between the third nucleotide of the anticodon and the first nucleotide of the codon is not always ambiguous. As of 8.5, the table, mutations that alter the hydrophobicity of the amino acid are less likely to be determinantal of the protein function. The average difference in the hydrophobicity upon the replacement of a nucleotide in a codon varies depending on the position of the nucleotides. So, the second topic begins. 8.8, regulation of genes. So, in this section, divides into two intricate process of gene regulation in living organisms. It emphasizes the protein biosynthesis. The process of creating proteins from the DNA is carefully controlled. Molecular recognition. The recognition of genes occurs at the molecular level through the interaction involving weak forces. These interactions are crucial for the entire biological development of a cell. Mutagen factors. Substances that can cause mutations in the DNA can disrupt protein synthesis. However, even mutagen substances can significantly influence gene activity. Induced enzyme synthesis. In some cases, the presence of specific substances can trigger the synthesis of the enzyme. For example, the addition of methylgalactoside to the E. coli bacteria can induces the synthesis of the enzyme of beta galactosidase constitutive mutants. Some mutant strains of bacteria continuously produces certain enzymes even in the absence of the induce. Repressor and the inducer. So, what's repressor and inducer? A repressor protein synthesized by the regular gene binds to a operator gene and prevents the transcription of the structural genes. An inducer can bind to the repressor, inactivating it and allowing transcription to proceed. Lack operon. Lack operon, we have heard it lots and lots of time in the biophysics as well as physiology. So, what's lack operon? The lack operon in E. coli is well studied example of gene regulation. It controls the synthesis of the enzymes involved in lactose metabolism. The operon includes regulatory gene, which produces the repressor protein. Operator gene controls the access of the RNA polymerase to the structural gene. Structural gene encode the enzyme beta galactosidase and the beta galactoside permase. Complex genome structure. Eukaryotic genomes contain repetitive sequences, invented genes, palindromes, and the both functional and non-functional DNA segments, introns and the exons. Within functional genes, there are interesting non-functional sequences called introns, which are removed during RNA processing. So, this topic also comprises of transcriptions, acceptors and structural zones, pro-mRNA processing, regulatory proteins, chromatin structure, and fine-tuned regulation. So, the comparison with the prokaryotic regulation, the complexity due to the longer genome size and the presence of the introns, chromatin structure less prominent in prokaryotics, multiple regulatory levels including transcription, RNA processing, and translation. So that's all for this topic. Thank you so much for listening. Have a good day.